Hi, I'm Helen Willsey. I'm an assistant professor at UCSF and an investigator with the Chan Zuckerberg Biohub. I'm sorry that I'm coming to you remotely, but it is most likely like 7 a.m. where I live, and so I am probably hurrying to get children out the door to school. But today I'm going to tell you about what my lab has started working on in the context of KCNT1 and specifically using frogs to study a potential role in cilia biology. So my lab studies hundreds of different genes. We're really interested in ones that are associated with rare disease and carry large effects. So we're usually studying autism genes, but we also study a lot of genes that carry risk for epilepsy and particularly pediatric epilepsies. To study this many genes in parallel, we use a high throughput vertebrate model, which are frogs, of course. And so specifically, I'm talking about Xenopus tropicalis. And these are diploid frogs, and one mating pair can produce over 4,000 embryos in a day. So this really lets us do genomics at scale, studying many different genes in parallel and understanding their underlying biology. Frogs are very cheap to have in the laboratory, and they develop very quickly. So by 5 days of development, they're eating, swimming, behaving, circadian rhythms, all that. And genetic manipulation is really nice in frogs. You can introduce mutations by CRISPR-Cas9 and so on and so forth. And drug screening is also high throughput because you can just put the drugs in the water and the embryos can be in 96-well plate format. So it's a nice system to study many genes in parallel. And one of the things that we have discovered studying autism genes is that many of these genes, which are also associated with rare epilepsies and pediatric epilepsies, a lot of these same proteins show up on a structure called the cilium. So I'll give an introduction to the cilium in a second. But these are proteins that were previously thought to only be present on the synapse. So much so that associated disorders were called synaptopathies. But here what we've shown is that actually these proteins also localize to the subcellular organelle called the cilium. This work I'm showing you here was done in frogs, where we take the human protein and we put it into a ciliated tissue, tag it with GFP, and see where it goes. And what you can see here is that for these classic synapse genes like SYNGAP, SHANK, SCN2A, they're really localizing to the base of the cilium as well as into the cilium structure itself. This is frogs. This is a kind of an interesting cell type to be looking at. And so here, what I'll actually show you is endogenous localization of one of these synapse genes, SYNGAP1, on the cilium marked by RL13B in a human iPSC-derived excitatory neuron. So we use the frog as a model, and then we test what we find in the frog in human-based models to make sure it still translates. In this case, this gene, which when varied can cause rare pediatric epilepsies, is here on the cilium. Let me tell you a little bit about the cilium for a second then. Why do we care about cilia? And so every cell in the body other than blood cells has a cilium, including neurons. So I just showed you that iPSC-derived excitatory neuron. Here's another one. And now I'm showing you this pink structure right here is very small, but that is the cilium on an excitatory cortical neuron. And they're thought to be the cell's antenna. And up until the 90s, they were thought to be vestigial structures carried on through evolution from algae. But actually, they're very functional, even in neurons, in sending and receiving signals. They signal through calcium. They do a lot of developmental signaling, like sonic hedgehog signaling, the pathway that's very important for the development of the pulmonary arteries. They're involved in neuronal transmission. A lot of dopamine and serotonin receptors are specifically localized on the cilium, as well as many GPCRs. And they also directly influence excitability of the neuron itself. So it seems to be not just the synapses, but also the cilium, where these proteins are functioning and having effects. And so it's known that when there are cilia problems in a person, this leads to a whole wide spectrum of issues, depending on where that protein is expressed and in which tissues it's controlling ciliation. But for this group, what I'll highlight is the is that cilia defects themselves can be a cause of pediatric epilepsy on its own. So I think it's worth exploring the possibility that KCNT1 has a role in ciliation. I also think it's worth exploring because there's precedent for other potassium channels to function at cilia. So for example, here's KCNH1, which of course is a very different potassium channel, but it has been shown to localize both to the base of the cilium and within the cilium, and overexpression and activation of this channel has shown to cause cilia defects. And so I think that raises the possibility that potassium flux at the base of the cilium is very important for controlling ciliation, and that other potassium channels may act similarly. So to test that hypothesis, we've done some very preliminary early experiments to think about this. And so what we did was we took human KCNT1 and tagged it with GFP and did what I showed you earlier and just put it in the frog and this big ciliated cell type and say, where does it go? And much like those synaptopathy proteins, what we see is it localizing to what's called the basal body, which is the very base of the cilium that organizes the cilium out of it. And then also faintly along the cilia as well. And when we overexpress this reference sequence of the protein, we get a very interesting cilia phenotype, which is you get these bulbous tips to the cilia at the end of them. And so we think that these are exciting early results that are now motivating our future work to test the hypothesis that KCNT1 is functional in ciliation, and that this function may be relevant to the patient condition and could even be therapeutically actionable. So I have a wonderful new graduate student who has taken on this project, Angeline. And if people want to chat, I would love to collaborate and think about how we can study KCNT1 on cilia. And so my email address, that's the best way to get a hold of me, is just my name at ucsf.edu. So please reach out. And if you want to chat cilia or frogs or KCNT1, I'm more than happy to. So thanks for your attention. Talk to you later.